Over 130 years ago, a Catholic monk named Gregor Mendel discovered some basic laws of heredity. Mendel worked with pea plants, much like these, here at the University of Wisconsin Research Lab. And the laws he discovered about peas, it turns out, work equally well for Brussels sprouts, tomatoes, apple trees, and human beings. Well, today in labs all over the world, thousands of workers are building on Mendel's laws of heredity, finding new ones, and creating a revolution in life science. I'm Bill Stonebarger, producer of the program you're about to see, a program about the most incredible assemblage of molecules in this world, an assemblage called the gene. Monsters have a long and noble heritage. For centuries, people believed that giraffes came from the mating of a camel and a leopard. If the camel mated with a sparrow instead, an ostrich was born. In Russia, people thought that very strong men sometimes came from a woman being kidnapped by a bear and conceiving child. Now, besides the monsters people believed in, but no one had actually seen, there were genuine monsters people had seen. In the Renaissance, particularly, there was a great interest in freaks of nature, as they were called, two-headed calves, Siamese twins, babies with horns, and people with gross deformities. Separating the make-believe monsters from the real monsters was difficult because no one knew much about heredity. That is, just how life's forms were passed on from parent to offspring. From very early times, people understood that sex was needed to produce offspring in the higher animals. They didn't think it was necessary for lower creatures, however. Aristotle, for instance, the Greek philosopher and scientist, who was considered the greatest authority on science for over 1,000 years, said that worms and flies and such were spontaneously generated out of muck and slime. No sex needed, no parents required. Even when parents were obviously involved, no one had any idea what it was that made offspring resemble their parents, nor what it was that made them a little different, too. In other words, no one had any idea how heredity worked. Today, we know how heredity works. We know what it is that makes offspring resemble their parents. It is genes. Well, this is the story of how humans found out about genes and how genes work in our living world today. Now, the first real scientific insight into heredity came 140 years ago, about the time of the American Civil War, in a now abandoned garden in Brunn, Czechoslovakia. The breakthrough was made by an eccentric Catholic monk named Gregor Mendel. Mendel was a busy man. He smoked 20 cigars a day, kept 50 hives of bees and many cages of mice. He was regional weather correspondent for the Austrian Empire. He was elected abbot of his monastery, and he carefully nurtured and studied thousands of garden pea plants. For this last task, the world is forever in his debt. See, up until Mendel's time, even educated people thought that inheritance was somehow carried by the blood. They thought that offspring were a blend of the qualities of their parents. They even thought that if a man had a tattoo on his right arm, his child might also be born with a tattoo on his right arm. Mendel's work with garden peas proved all these beliefs to be mistaken. Inheritance, he showed, was not carried by blood or any other general body cells of plant or animal. Instead, it was carried by some kind of special something that was hidden in the sexual cells, the egg and the sperm cells. These special somethings were later given the name genes. Well, here's how Mendel did it. In his monastery garden, he experimented. He took the male cells of peas that produced tall stem plants, for instance, and crossed them with the female cells of peas that produced short stem plants. To his surprise, from this cross, Mendel ended up not with a blend of tall and short stems, but with all tall stems. 100% tall stems. Well, what had happened to the short stem trait? It had not blended. Had it been lost? Mendel did another experiment. He took the tall plants that he got from crossing tall and short ones, called now the hybrid first-generation plants, and he crossed them with each other. Lo and behold, this time, the second generation, he did get some short-stem plants. In fact, on the average, 1/4 of the new pea plants turned out to be short-stemmed ones, and 3/4 turned out to be tall. And Mendel got these same ratios when he tried 6 other simple characteristics: wrinkled and smooth seeds, axial and terminal flower buds, green and yellow seeds, yellow and green cotyledons, inflated and constricted pod shapes, and gray and white seed coats. Well, some thought Mendel was a little odd to spend so much time counting seeds, and when he published his results in the Proceedings of the Balloon Society in 1866, it did not make much of a splash. In fact, it was not until 40 years later, long after Mendel was dead, that the scientific world did take notice. And today, all biologists agree that Mendel's discoveries are indeed basic laws of heredity, valid not only for garden peas, but for elephants, for worms, for swordfish, and for humans. After Mendel, progress was still slow in learning more details of heredity, and there was backsliding. Despite Mendel's work, the great pioneer of evolution theory, Charles Darwin, still believed in blending. He also thought that traits picked up in the lifetime of plants and animals could be inherited through what he called gemmules in the body cells of plants and animals. As late as 1899, a student of biology at Brünn found Mendel's 1866 paper and eagerly showed it to his professor. "Oh, I know all about that paper. It's of no importance," said the professor. "It's pure Pythagorean stuff. Don't waste your time on it." See, one of the problems was that investigators were too ambitious. People like Francis Galton in England, for instance, carried out scores of ingenious studies on the inheritance of complex human traits like intelligence, size, and bodily strength. Galton even tried to develop a numerical scale for beauty and love, and once did a statistical study of disasters on ships that carried missionaries versus those that did not. All of these complex traits and happenings we know today are due to a bewildering combination of thousands of genes and even more thousands of environmental factors. Around the turn of the century, botanists were independently finding Mendel's laws to be true. One far-seeing German scientist, August Weismann, was already predicting in 1885 that heredity would eventually be found to be carried by definite chemical and, above all, molecular structures. It remained for the 20th century to discover just what that molecular structure is. The most dramatic breakthrough in heredity since Mendel's garden happened here along the banks of the Cam River in Cambridge, England in the 1950s. A quiet young American biologist and former quiz kid from Chicago, James Watson, used to stroll along this path with an English biologist in his mid-30s, famous for his loud voice and even louder laugh, Francis Crick. Crick said of Watson he was the first person I had met who thought the same way about biology as I did. And Watson said of Crick he was the brightest person I had ever worked with. Along this path, in a tiny office at nearby Cavendish Lab, and over long luncheons at the Eagle Pub, Crick and Watson puzzled about what genes were and how they worked. The place, the time, the spirit, all were ready, and the sparks from the combination led to the greatest revolution in biology since that of Charles Darwin's theory of natural selection 100 years before. You see, by the 1950s, much more was known about cells and about the Mendelian laws of heredity. T.H. Morgan at Columbia University and Herman Muller at Indiana University had used fruit flies to locate genes on the chromosome. They had shown how Mendelian laws still apply, but in a more sophisticated way than Mendel imagined. Muller, in particular, had shown how X-rays can cause mutations, that is, permanent changes on a chromosome or on a gene. He was able to construct full-scale maps of the genes on the chromosomes of a fruit fly. A few decades later, geneticists would be able to do the same with human genes and chromosomes. One of the most important keys in this progress was new and better scientific equipment. Microscopes, for instance, have become much more powerful. Using new X-ray diffraction techniques, scientists could peek into the actual molecular structure of large molecules. And new chemical techniques were invented: chromatography, electrophoresis, radioactive tracing, that could separate and identify unbelievably tiny amounts of complex chemicals. But each one of these advances required the thought and work of many hundreds of researchers, so that it is misleading and unfair to single out one or two people to get all the credit. So too with the discovery of the molecular structure of genes, that is now usually credited to the famous Crick and Watson team. The particular tools that Watson and Crick used to make their breakthrough were model building, some new X-ray data, and inspired guesswork. The principles needed for the model building had been worked out across an ocean and a continent by the chemist Linus Pauling at the California Institute of Technology at Pasadena. The X-ray data had come from the work of two scientists in London, Rosalind Franklin and Maurice Wilkins. The guesswork was Watson and Crick's. Later, 3 of the 4 received the Nobel Prize for the brilliant answer they gave to the question, what is a gene and how does it work? Sadly, the 4th, Rosalind Franklin, died a premature death from cancer in 1958, 4 years before the Nobel Prizes were awarded. Well, what exactly did Crick and Watson do? Well, they figured out the architecture of a very large molecule, deoxyribonucleic acid, DNA for short. This molecular structure was so important because it is DNA in the nucleus of all cells that is the actual physical thing that carries the all-important information of heredity. The special something that Mendel had assumed in pea plants, the molecule that Weissman had predicted in 1885, and the gene that Muller had mapped on fruit fly chromosomes. DNA is the physical molecule that carries the computer-like code that tells your cells how to make an eye, an arm, a brain. And not just any eye, any arm, any brain, but your eye, your arm, and your brain. Not just you, but your dog, your goldfish, your poison ivy, your earthworm, the largest whale in our ocean, and the smallest virus in our bloodstream. All living things, in other words, are built and operated following instructions from the DNA in their cells. And Crick and Watson could now diagram and make models of the very most secret heart of life itself. Well, here is the actual diagram Crick and Watson drew for their famous paper, A Structure for Deoxyribose Nucleic Acid, published in the scientific journal Nature, April 25, 1953. DNA, they guessed correctly, was a double helix structure. The helix backbone was always the same, a well-known chemical group called phosphate sugar. Connecting the 2 phosphate sugar ribbons are pairs of 4 well-known chemical bases called guanine, cytosine, adenine, and thymine. To a chemist, it was simplicity itself, and an elegant, beautiful simplicity, especially when they saw that guanine can only connect to cytosine, and adenine can only connect to thymine. Yet, despite that limitation, the helix is long enough so that an almost infinite number of possible sequences is possible, which means, of course, that an almost infinite variety of life forms is possible. And finally, as Crick and Watson said in their famous paper, it has not escaped our attention how this double helix structure provides powerful hints as to its method of replication within the cell. Well, in part 2 of this program, we will show how simply that replication works. Again, an elegant, beautiful solution to an age-old puzzle. Well, just finding the actual molecular structure of DNA did not in itself solve all the problems of genetics. It did provide, however, the most powerful tool yet found to investigate these problems. Now scientists could isolate the parts of the cell that were the sites of the actual physical genes. They could proceed to take these genes apart and put them back together again, in old ways and in new ways. And today, in laboratories all over the world, geneticists and biochemists are making rapid progress in deciphering life's codes in plants and animals, including humans. Genes have been spliced from one organism to another. Genes have been repaired. Synthetic genes have been made. Genes have been mutated. Genetic surgery is now widely used commercially as hundreds of new biotech companies are launched. Whole new kinds of life forms, especially new strains of bacteria, are being designed and made in the laboratory. New kinds of life forms engineered to do specific tasks like create hormones, antibiotics, vaccines, mine metals, clean up pollution, even build computers. And in one of the biggest scientific projects of all time, geneticists are right now mapping all the genes of the human species. This project is being led, fittingly enough, by James Watson himself. You know, the past 50 years have led to a revolution in biology. The next 50 years will have their surprises. Well, now that we've seen how the gene was discovered, let's look in part 2 of how the gene works and how knowledge of the gene can help unlock more of life's secrets today and benefit all life on Earth, including human beings. Alice laughed. "There's no use trying," she said. "One cannot believe impossible things." "I dare say you haven't had much practice," said the Queen. "When I was your age, I always did it for half an hour a day. Why, sometimes I've believed in as many as 6 impossible things before breakfast." And if Alice could wander through the wonderland of modern genetics, she would need even more practice. For here at the very heart of life, the impossible is an everyday thing. Would you believe, for instance, that less than a teaspoon of a substance called DNA, deoxyribonucleic acid, would be enough to recreate all the people now living in this world? Or how about this? Before the end of the century, we may be able to actually recreate you from the information coded in the DNA of any one of your cells, say one microscopic cell from your big toe. Right now, today, we can transfer a gene from one plant to another and from one animal to another. We can make a synthetic gene for rabbit hemoglobin. We can create new kinds of living bacteria. And some of these bacteria can mine copper for us, can make insulin and other hormones, can produce new antibiotics, can fix nitrogen into fertilizers, and someday soon may even be able to produce and reproduce living computers. Now, all of this is not science fiction. Scores of hard-nosed business people have invested millions of dollars in new high-tech companies springing up all over the world to make these impossibles everyday realities. So let's look at the basic science that makes all this possible. Just as knowledge of the atom is the most important key to understanding physics and chemistry today, well, knowledge of the gene is the most important key to understanding biology today. So what is a gene? Gene, we know today, is a molecular structure that carries the code needed to govern life functions. Just as a computer needs a program, so a cell and a complex combination of cells called an organism needs a program. That program is in code, and the code is on a spiraling molecular structure called deoxyribonucleic acid, DNA for short. A molecule of DNA actually carries more than one instruction, more than one gene. Along this spiraling DNA molecule, for instance, one section might be part of the code that, in interaction with other genes, governs eye color. Another section could have a code needed to produce the hormone insulin. Another section would have a code for hemoglobin in your red blood cells. Obviously, in order to create an entire person, you need a lot of genes. Between 50 and 100,000 is the best current estimate. And these genes have complex interactions as well, many billions. The possible combinations get astronomical. How else could we have such a rich variety of living things in our world? When you think of all the possible combinations of magnetic pulses on a tape, grooves on a record, words in a book, DNA is like that, only more so. More information and more power. Now, the DNA is surrounded by protective protein coats and is strung like beads along structures called chromosomes. These chromosomes wind around in every cell's nucleus. The DNA in each chromosome has thousands of genes. One surprise is that much of the DNA does not seem to carry any coded messages at all. We don't know at this time why this should be so. Perhaps nature, like human beings, can be redundant and wasteful, as well as efficient and powerful. Just about every cell in the living body of every organism has a store of DNA in its nucleus. The same store as in every other cell of the same individual organism, and that's why cloning is possible. Cloning has been known and practiced for many years by gardeners, but recently it has been done experimentally with animals too, including mammals. Cloning is a way to use the coded instructions of a general body cell to reproduce the whole organism without sexual recombination. Thus, the cloned individual turns out to be exactly like the parent in every respect, since he or she has exactly the same genetic instructions within each and every cell. Normally, of course, this is not the way organisms reproduce. Normally, the genes that are going to be used to create new organisms are very early along segregated from other body cells. They're kept separate in the ovaries of the female, the eggs, and in the testes of the male, the sperms. Because they are segregated so early, experience of the adult organism has nothing to do with the heredity given to the offspring. Thus, inheritance of acquired characteristics is not possible. When it comes time to reproduce, cells in the testes and in the ovaries go through a special kind of division called meiosis. In the diagram, you see a microscopic view of what happens to the chromosomes in meiosis. They divide in two. Each sperm and each egg thus gets 1/2 the regular number of chromosomes and genes. In your case, for instance, this means you got 1/2 your inheritance from your mother and 1/2 from your father. When your parents had sexual intercourse, these half-number cells joined together to make a new whole-number cell, a fertilized egg. The egg from your mother and the sperm from your father, in other words, fused together to make one complete cell, a fertilized egg cell, that in a few short months was born as a unique new person, you. In those few short months came the impossible part. The part we still know so little about it seems near miraculous. The single fertilized egg cell had all the codes needed to create you. To let these codes do their job, all the fertilized egg cell needed was the right environment. Your mother's uterus provided that environment. It provided the right temperature and the right chemicals. It provided a salt-watered world very similar to the ancient oceans, where amino acids, sugars, salts, and fatty acids are abundant and ready for use. When all is right, the process unfolds like the most dramatic motion picture you have ever seen. The single fertilized egg cell quivers, doubles its chromosomes, and divides into two. This is called mitosis. Notice it is different from meiosis in that the chromosomes, and thus the DNA and the genes, doubled before the cell divided into two. Thus, each new cell has a full set of chromosomes, DNA, and genes. Let's look at this doubling more closely. Remember how Crick and Watson put it in their epoch-making article? It has not escaped our attention that this double helix structure provides powerful hints as to methods of replication within the cell. Well, here's what they meant, and here's how it does work. The double backbone of the helix is made of phosphate sugar groups. These are connected, like rungs on a twisted ladder, by 4 different kinds of chemical bases: guanine, thymine, cytosine, and adenine. Well, guanine, it turns out, can only hook itself to cytosine, and adenine can only hook to thymine. That is sufficient, though, to create an intricate code and to provide an ingenious way of replicating. With the help of other important chemical molecules called enzymes, the 2 backbone parts of the DNA molecule untwist, and then the whole molecule unzips. This gives 2 half DNA molecules, but 2 halves with highly active base parts. See, now here's where the environment comes in again. Floating in that saltwater soup are plenty of guanine, cytosine, adenine, and thymine groups. Where'd they come from? Well, from the food that the mother ate and supplied to the uterine environment. With the help of still other important enzymes, each half of DNA hooks onto the base group as needed. That is, every guanine hooks onto a cytosine, every adenine to a thymine, every cytosine to a guanine, and every thymine to an adenine. Phosphate sugars are also nearby, and so we zip back up, and now we have 2 DNAs, where there was only 1 before, and 2 identical DNAs. The cell now divides, the chromosomes divide, the DNAs divide, and you have 2 complete cells with 2 complete sets of code. Each cell identical to the one it came from. And the process continues. 2 become 4, 4 become 8, 8 becomes 16, 16, 32, and so forth, and so forth, and so forth. And we get millions and billions of cells, all with the complete set of chromosomes, DNAs, and codes needed to make you, you. But wait, how come the cells are not all alike then? The cells in your eye, for example, are not like the cells in your ear. The cells in your lungs are not like the cells in your biceps. How did they all become different? For the most part, that is still a great mystery. No one knows, not yet. This great mystery is given a name. It's called differentiation. Now, there's a lot we don't know about differentiation. We do know, though, that it's governed by the same genetic code that's on the DNA molecules. And we know a little of how the code is transmitted from DNA in the nucleus to the rest of the cell. The DNA is able to pass along its coded messages to a very similar single-chain molecule called ribonucleic acid, RNA. This smaller RNA molecule is able to slip out of the nucleus and into the rest of the cell. And there it takes charge of the building of new enzymes and proteins. And finally, it is these enzymes and proteins that do the actual work of the cell. Carrying oxygen, for instance, if you happen to be a red blood cell. Carrying electrochemical pulses if you happen to be a nerve cell. Contracting if you happen to be a muscle cell. Amazing. Amazing not only that it works, but even more amazing that scientists have been able to figure out how it works. Of course, it's one thing to do this in theory and another to do it in practice. Today, there are many genetics laboratories all over the world cooperating and competing to push forward our basic knowledge of the gene. The tools of all these scientists are similar. Some of the most basic are as old and common as any in science. Test tubes, beakers, sterile culture dishes, pipettes, colored tapes, a variety of chemical reagents, Bunsen burners, and refrigerators. Others are newer and more sophisticated. Electron microscopes, electrophoresis setups, scintillation counters, ultra-high-speed centrifuges, and, of course, ultra-fast computers. The techniques worked out to probe these submicroscopic genes are ingenious, to say the least. For instance, suppose you want to cut a strand of DNA at a particular spot, say where the gene for eye color is located. No problem. Today there is a large variety of chemicals called restriction enzymes that can act as sharp and ever so accurate chemical scissors. You can buy these restriction enzymes off the shelf. Other chemicals, usually radioactive tracers, are used to mark the points of cutting. Then various gels can be used in combination with electrical charges to separate the DNA fragments. Once separated on a piece of paper or a gel, a computer is brought in to help interpret the patterns found. And the final result is a photographic record of the sequences of guanine, thymine, cytosine, and adenine. Whether on film or reprinted on paper, the sequences are very long. Geneticists today are a little like a child just beginning to read. They're still at the primer level. What will it be like when we reach the length and sophistication of a War and Peace length novel? Okay, it's time to summarize the basic facts before we take a look at the controversial future, the engineering of new life forms. 1, the gene is the biological unit that carries heredity in every cell of every living organism, from viruses to fruit flies, from pea plants to people. 2, the gene itself is a molecular structure, a group of atoms that are part of a large molecule called deoxyribonucleic acid, DNA. 3, it is the pattern of base sequences on the DNA that is the code, which when translated by living cells make a virus, a fruit fly, a pea plant, and a human. Not only in general, but a particular virus, a particular fruit fly, a particular pea plant, a particular human being. 4, when you change the structure of the DNA sequence, you change the code. You change the living tissue, you change the living organism. It's like changing the computer program or the words in a book. You end up with a new program, a new book. Maybe better, maybe worse. In the case of genes in nature, the new living organism that results from a change in the gene, it's called a mutation, is usually worse off than its parent. 5, very occasionally a mutation may lead to a new trait in the offspring that is to its advantage. That offspring will have a better chance to survive and pass on that new mutant gene to its offspring. And that's the way change and natural selections occur. Well, what does this all mean in practical life today? For one thing, today humans are trying to engineer genetic changes for the good. And they do this by using what is called recombinant DNA technology. Say you find a gene that is capable of some desired task, like cleaning up oil spills, producing insulin, gobbling up some nasty toxic waste, fixing nitrogen in a fertilizer, or whatever. Using one of the new restriction enzymes, you snip it out of the cell it is found in normally, and then you paste it into an opened up cell in a fast reproducing bacteria. You give the newly enhanced bacteria enough food and living space and let it reproduce. Within a very short time, you have billions on billions of these powerful new living things, new living factories. You purify the substances they have produced, bottle them, and sell them to people who need them. Similar techniques are also being developed to cut and paste genes from one organism to another, say from a bean plant into a corn plant, or from a bacteria into a rabbit. As always with new knowledge, there are risks. What if some of the new human-created genes escape the factory and laboratory and play havoc with natural genes, our own or other natural creatures in the web of life? Will the imaginary monsters of the past become only too real this time? And will the real monsters multiply beyond our control and our worst nightmares become reality? Or will we enter a new era where our farms and orchards are more fruitful, our factories and our mines safer and more productive, our cities and our countryside freed of pollution, and all of our people, all the world's people, freed from inherited diseases, free to live longer, healthier, and happier lives? All of these possibilities and more are being actively studied and debated today. And now that you understand the basic principles of this new genetics, you too can take a role in the debate. For in a democracy like ours, how this immense new power is used will be up to the most important people, you and me.